Welcome to the Learn Biology podcast, where we break down complex biological concepts and make them easy to understand. Today, we're diving into one of the most fundamental processes on Earth, photosynthesis. Get ready to explore how plants and other photosynthetic organisms convert light into life. Photosynthesis is the incredible process that allows plants, algae, and some bacteria to transform light energy into chemical energy stored in the form of glucose, the fuel of life. The overall reaction for photosynthesis can be summarized as carbon dioxide plus water plus light energy produces glucose plus oxygen. Let's first start by reviewing some key terms and concepts. Pigments are the molecules that capture light energy during photosynthesis. The most essential pigments are chlorophyll A, chlorophyll B, and carotenoids. Accessory pigments are used in addition to chlorophyll, and they help cells by allowing them to absorb additional wavelengths of light. They also help cells neutralize harmful free radicals which would damage the cell. Next are photons, which are particles of light that serve as packets of electromagnetic energy. The absorption spectrum is a curve that illustrates which wavelengths of light a pigment absorbs most effectively. The action spectrum indicates if photosynthesis occurs when a specific wavelength is absorbed. Now let's discuss some of the cellular machinery that carries out photosynthesis. First, we have photosystems. These are complexes of pigments and proteins within the thylakoid membrane that are essential for light absorption. Within the photosystem, the antenna complex captures photons and channels their energy to the reaction center, which will drive the start of the photosynthesis light-dependent reactions. Photosynthesis takes part in two major sets of reactions. The first set are called the light-dependent reactions. These reactions start at the photosystems and use sunlight to generate the production of ADP and an electron carrier called NADPH. Both of these products are required for the cell to complete the important part of photosynthesis called the carbon fixation steps or the Calvin cycle. In these second set of reactions, the cell uses the ADP and NADPH produced in the light-dependent reactions to link CO2 together to make sugar for the cells. Let's now describe the light-dependent reactions in a little more detail. The first stage of photosynthesis occurs in the thylakoid membranes. There are two types of photophosphorylation that occur. Remember that photophosphorylation refers to using the energy of photons to add a phosphate to adenosine diphosphate to make ADP. In non-cyclic photophosphorylation, a photo is absorbed by a photosystem and the energy is channeled to the reaction center. There, an electron is lost from a chlorophyll and it moves through an electron transport system to make ADP. The electron continues on to a second photosystem. There, another photon is absorbed and channeled to the reaction center. This time an electron is relayed through NADP reductase, which is an enzyme that produces the electron carrier NADPH. Lastly, in order to keep the first photosystem replenished with electrons, a water molecule is split, which releases an electron for the process to start over and generates the oxygen that is released by plants. To recap, the main products of non-cyclic photophosphorylation are ADP, NADPH, and oxygen. Remember, the plant needs ADP and NADPH to make sugar later in photosynthesis. The second type of photophosphorylation is called cyclic photophosphorylation. In this step, photons are absorbed by a photosystem and relayed to the reaction center. There, an electron is sent off through an electron transport chain to make ADP. The electron then returns to the same type of photosystem, completing a cycle. In cyclic photophosphorylation, only ADP is produced. This provides even more additional ADP to supplement the ADP produced in non-cyclic photophosphorylation. The second stage of photosynthesis is called the light-independent reactions or Calvin cycle. These reactions take place in the stroma of the chloroplast and involves carbon fixation, which is the process of carbon dioxide being taken out of the atmosphere and added to organic molecule called RuBP to make PGI, which is the start of the sugar-making process. Rubisco is the enzyme that does this very important step in photosynthesis. The next step is called reduction. The PGA molecule generated in the first step is reduced, which means electrons are added to it. This is completed using the ADP and NADPH from the light-dependent reactions. The end molecule is called glyceraldehyde 3-phosphate or G3P. The last step of the Calvin cycle is the regeneration of RuBP. This is required to keep the cycle going. Overall, the Calvin cycle uses the ADP and NADPH from the light-independent reactions to perform carbon fixation, which leads to the production of G3P. This molecule will go on to be used to make sugar for the plant. And there you have it, a detailed look at the remarkable process of photosynthesis. From capturing photons to generating glucose, photosynthesis is a stunning example of nature's ingenuity. It not only provides energy for plants but also sustains all life on Earth by producing the oxygen we breathe. That's all for today's episode of the Learn Biology podcast. Don't forget to subscribe and leave a review. See you next time.